Good morning, everyone. It's probably now time to start. Good morning. My name is Francesco Petruccione, and I'm the Pro Vice Chancellor of Big Data and Informatics at UKZN. And if I'm allowed to do a little advert, since a few days, I'm also the interim director of what will become the National Institute for Theoretical and Computational Science. Welcome to this second edition of the online edition of the Data at Breakfast. Yeah, before I introduce the speaker, I just want to to share some rules of the webinar. Yeah. Unfortunately, none of you will be able to directly speak to the speaker and ask directly questions, but there is on the bottom bar of your Zoom screen, there is a question and answer button, yeah, where you're welcome to write pose questions, and either Tulio will browse the questions while he talks and directly answer to you, or otherwise I will try to summarize your questions and pose them to Tulio towards the end of the talk. Yeah. Okay, let me introduce the speaker now. Professor Tulio de Oliveira is the director of KRISP, which is based at the Nelson Mandela School of Medicine of UKZN in Durban. He's also an associate professor at the Department of Global Health at the University of Washington and a senior research fellow at CAPRISA. He's a trained bioinformatician and has published more than 150 papers, including a very recent bioinformatics tool for genomics analysis of coronavirus genomes. Yeah? And Tulio is one of the driving forces behind the COVID-19 data analysis and reporting efforts at UKZN. Okay, Tulio, now I invite you to start your presentation, and you're welcome to share your screen so that everybody can see your slides. Thank you very much for agreeing to give us a talk this morning.
Hello, everyone. Yeah, good morning, and great to see so many people come to the talk. Yeah. So today, what I'm going to be talking to you, it's about COVID-19, a little bit about the epidemiology, but more about the genomics. What I'm going to try to highlight to you, it's how important this new area called genomics, that means studies of genomes, have been to really understand and elucidate transmission and origins of COVID-19. So if I go to my next slide, it is always good to start with a nomenclature. Yeah. So everyone understand about COVID-19, that's the name of the disease. But what they also have done is to identify the specific virus, which called severe acute respiratory syndrome related coronavirus 2, or in short, SARS-CoV-2. And that will be important because as I show you during that presentation, you're going to see how similar this virus was from the SARS outbreaks that happened around 20 years ago. So if I go to the next slide, and I start here with last week's slide of a colleague of mine, Richard Lassos, that gave this talk here. Yeah. And a week ago, we had 520,000 confirmed cases in 175 countries and 25,000 deaths. A larger number of cases and deaths. But if I go to what's happened today, we have over 1 million confirmed cases and more than 50,000 deaths. So the speed of this pandemic is really unprecedented. Yeah? In less than a week, yeah, or basically in a week, yeah, you double the number of cases and you come to that number that we never wanted to go, that's above the millions. If I go to the next slide, of course, people say, okay, that's around the world, but what it is in South Africa? And you may have seen this slide last week. So that's part of the group that we work with Francesco and a lot of his researchers and our researchers to try to map in real time how South Africa COVID-19 epidemic is unfolding. Yeah? So that slide, I just want to, it is a statistics that we do the rate of growth And what you see that as last week, South Africa was growing at the same rate as all the worst European epidemics, such as the Spain or Italy. If I go again to this week, what we're going to see is that it's quite a different scenario. In the last 7 days, we see South Africa decreasing the rate of the growth. And if we put a little bit more in details of the work that we do together, and this graph is from Maria Schuder, and I'm gonna take a minute to explain to you. Here on the top left, we compare the rate of growth of South Africa with some of the worst epidemics in the world. So as I say, last week was similar, it starts to deviate. Here to the right, it is the growth of South Africa, but now with the countries that control very well, such as Singapore, Japan, and South Korea. So South Africa may be going the same area. It may be too early to see, but it's quite good to see these results. In the bottom in purple, we have South Africa compared with two countries like Poland and Romania that did very similar South Africa, very strict, yet very strong response, lockdown very early. And South Africa is growing at the same rate of them. What it means at the moment, the epidemic is doubling every 4 days, or less than 25% growth per day. And the last graphic here, South Africa compared with the southern countries, two southern hemisphere countries, big ones like Brazil and Australia, because a lot of the hypothesis is that we would be in summer or spring, but South Africa now is beginning to grow slower than that. So that's very interesting results. We are just finishing a paper together with Francesco and our team. We're gonna be submitting today and making available today. But what I want to really talk to you, it is how fast this all happened. So I just have a slide here that talked 3 months ago. Only 3 months ago, to be more specific, on the 3rd of December of 2019, a first case of unknown pneumonia was identified in China. And in 3 months' time, we have isolated the virus. Here is an electrical microscope picture of the virus, which was initially called 2019 novel coronavirus. Now it's renamed as SARS-CoV-2. We have isolated the virus. We have understand all the 3-dimension structure. We have identified every single protein of the virus. The speed of science has been phenomenal on this outbreak. If I go to the next picture, is what we call a genome. And that may be quite difficult for many of you, but what I'm trying to say, and the only take home from this slide, is that we understand very well the genome of the virus. We understand every single protein, and the difference between SARS coronavirus 2 and SARS is just one extra tiny little protein, potential protein number 8. Otherwise, the genome is the same structure with the same proteins with the same function. So if I go to the next slide, you can get more in detail of the genome. It's the same genome in another graphical representation. The genome of the virus has 30,000 nucleotides. Just to compare with humans, humans have 4.5 billion nucleotides. So how tiny the genome it is. And one protein that we have been very interested as a scientific community is this protein called a spike protein. That's the proteins that outside the virus that make the crown, which bind in the human receptor to infect especially lungs. So we really understand what it is, the positions of these proteins that will bind to the human cells to enter. The receptor in the human cells called AC2. And there is also a small difference on that proteins that the human SARS coronavirus, the top one, it has a few introductions of amino acids which create an extra cleavage site, which is believed to give further transmissibility to this strain as compared with the other SARS coronavirus. So was phenomenal amount of information that you learned from that in the last few months. In the next slide, the genomics was also used to understand the origin of the virus. And again, this may be complicated to some of you, but if you look to the left on this phylogenetic tree, what you're going to see is that the first virus that was called the Wuhan human 1 virus, yeah, it is a subcluster of coronavirus called sarbecovirus. And if you look around in purple, you have another virus there that's called a bat coronavirus. And if you go down in that phylogenetic tree, you're gonna see bats, 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 and bats until you come to the SARS coronavirus. So what it means that the sarbecovirus, normally the natural reservoir is bats, and sometimes is transmitted to humans. We know that at least 2 times. That's the SARS, what would be SARS-1 now, and SARS-2. But what we know it is that this virus, the event to human, happened only once. And then after that, if I bring another phylogenetic tree, and that is from a family case of a household in Wuhan, is that after that, all the transmissions done by humans to humans. So just to summarize, that is a very rare event that the virus jumped from the natural reservoir from humans. And then after that, 99.99999% of the transmission will be between human to humans in close contacts. The same process happened with introductions of the virus across different countries. And here is a picture that it is, I'm gonna show more new information in the end of this presentation, but show that the virus originated in China and was very quick introduced to many countries in the world. A very important information here is the same with bat to humans. You have a few introductions to a given country, and then after that, 99% of the transmissions are with individuals in the same country. So one thing that we like to say is that almost doesn't matter where the virus comes from. What really matters is the response of your own country, which so far it seems that the very strong response in South Africa is beginning to pay off. Maybe too early to see, but some encouraging signs. If I go to the next slide, that's a very interesting slide. I'm gonna try to take you a little bit slower to that, yeah. Is that how you use genomic sequence to understand fine transmission chains and how that can be used to really control outbreak. So in the center of this diagram, you have here patient number 48. That's a patient that lives in Iceland that got in contact with a number of individuals. Some of them people from Iceland that have been into Italy, and other ones that have been to Austria. Yeah. And because what we did, or what these researchers did with the genome of the virus, they could first identify where the transmission come from. Was not from the individuals that have been to Austria, but through some of his friends that came from Italy that transmit to him, to close contact. But look what's happened. The chain of transmission of this individual within days in Iceland. It is involving another individuals, 68, 141, 107, 109, 104, 87, that then go cascading down. And now you can probably understand how it is important, the social distancing, to try to break these trains of transmissions. Because once the virus can infect one person, it can be spread very, very quick. So these individuals, 48, yeah, it transmitted to around 20 individuals within a few days as a source of cascades. And if you keep following that cascade without intervention, you can understand why you have exponential growth of the outbreak. So just to break a little bit to so much information, yeah, what fascinated us in the scientific world is how, when faced with a public health emergency, can research speed up to such a degree. So as I told you, it has been around 100 days since we first discovered the first case of pneumonia. In these 100 days, we have identified the virus, sequencing the genome, understanding transmission, understanding the proteins, understanding the receptor that enter in the cell. So how could you do that so quick? So one of the questions it is that the scientific community, and especially the Chinese in the beginning, decided to release data very, very quick. So what we are talking about from sample to scientific results in days. And we have like 6 main reasons for that. The first one is Twitter. I know that some people don't like the social media. Yeah, we are also not big fans, but Twitter has been the social media that the scientific community adopted and was the one that they discuss all the results. Another important point is open data. People is releasing data before they even analyze. Open software, open analysis. So you also release the results and the papers before they go to review. That's called preprints, and very fast reviews of the journals. So what I imagine is that that may completely change how we do science in the next few years. So we have participated in this process, and I'm gonna highlight here. And so I just decided to ground that on a piece of a Nature journalist, Jack Linning, that he did on our work and some colleagues on the beginning of the outbreak. So just to see how this kind of science was done very fast, but also involving multinational collaborations to do that very quick. That's a completely change from the traditional way that we do science. So for example, another piece on Nature a few years ago say that a paper takes an average of around 100 days to be accepted after submission. So imagine if you are in this process and someone managed to finish the paper and the result in the day that they identify the virus, what would be very unusual, you will still not know about that virus. So it's a completely end of the game that what in 100 days, we know so much. We have thousands of publications. So just change completely the process. If you go to us, for example, that's very normal. It's very normal in our research. We just recently got quite a very high impact paper that show how, and you show a 43% drop in the rate of HIV new infections. And that went to one of the big journals. But again, the data only went to 2017. So not only 100 days from publication, but what was common before this big outbreak is that we also take years to analyze the data. So let's see how the scientific community react to this and so fast. So that's just in some of the words of the Nature journalist, Jack Finn, talk about our work. We become involved on that very early. We become involved on the scientific research of coronavirus in the first week of January, when we saw some tweets from Jeremy Farrar, the director of the Wellcome Trust, and one of our colleagues, Andrew Raumbold, a molecular revolutionary scientist at University of Edinburgh. Yeah. So if I pass you on the 31st of December, one day after the identification of that virus, Jeremy Farrar was talking about how worrying a new virus have been discovered related to pneumonia that may be quite similar SARS, but highlighting how strong the China CDC is. And I think that's going to become quite clear how strong the China response was. Yeah. But that's as they have identified the virus. And then Andrew, as an evolutionary biologist, he followed that very close, yeah, identifying that the new report suggests viral pneumonia, no human-to-human transmission yet, linked to a fish market, patient, mainly business owners in the market, must be a repeated zoonosis from what? The linked location sound more like Legionella, but then why pneumonia? So the scientific community start really working together to tweet to try to understand that. If you go 2 days later, by 3 of January, as 8 of January 2020, a total of 44 patients with unexpected diagnosis of viral pneumonia was found. By 19 of January, we had 17 cases only. So, and that's how the scientific community reacts so quick. The main contact was through Twitter. Yeah. Quite interesting. One of the things that really helped to develop the science and understand the virus and transmission so quick was this post that's on the Virologica by Edward Holmes, another colleague in evolutionary biology, that released the first full genome the first full genome of this virus was released on 10th of January. Yeah, less than around 10 days after just diagnosing the first case. Really fast way, and was so important to have this first genome because then we could identify the virus, the source, how it is transmitted. Was really a crucial information to release that. Again, they released this data before they even analyze. And we become involved exactly on that day, yeah. So because our group developed a lot of bioinformatic tools and some of them to do phylogenetic analysis of virus, now we had a genome that we could start evaluating and creating a software application that could be used very quick to analyze of new of these genomes. So in the 22nd of February, we released a public and free version of a phylogenetic tool that could identify the new virus very quick and accurate directly from data from the sequences. So by the 2nd of February, the paper come online. Yeah, it was deposited, it was come online in the BioXV, one of the preprints, and really highlighting how important it was, the hand group, the release of the first genome for the whole scientific community, not only to respond, but to develop tools for that, yeah. So that was a collaboration that we did with a number of colleagues, both from Brazil and from Belgium, yeah. The paper was very well received. In less than a week, got like 2,000 accessions. Now it is over 5,000 accessions and has been used thousands of times to analyze different genomes. So how one can do something so quick, or how the scientific community reacts so quick, mostly was about us and other people being already studying virus outbreaks for a number of years. Yeah. So in our case, Genome Detective, our phylogenetic software, we already have worked quite a lot with a number of collaborators from Oxford and Brazil and Belgium in the last few years, yeah, to identify the source of yellow fever outbreaks in Brazil. That was a paper that was published in Science. Yeah, we also used that to identify the source of the Zika outbreaks in Brazil and the dengue and the chikungunya outbreaks. So the scientific community has been abnormal in the last 5 years, these new outbreaks of viruses, with the first big one was the Zika, and then chikungunya, yellow fever, and of course the biggest and most dangerous is the one that's happening now. So we had already systems that we could adapt it to that, but many of the other scientists that respond very quick, yeah. So what our software does, and what we also do in our laboratories here at UKZN in KRISP, is that we get the virus, we extract the DNA, we use these big DNA sequencers machines to break the DNA in thousands of times in pieces to sequence that very quick. And then our software Genome Detective will put a genome together, create a phylogenetic tree, identify the distinct virus, generate the genome, and then analyze all the mutations of that. And this whole process happened like in 10 minutes. So 10 minutes after you have a sequence, a virus being sequenced in the genome, you can release all the information back. So it's also very simple. You just copy and paste, or you load the original files in a web interface. The first thing that's going to do is to identify which virus it is identified. So in that case, this sequence was identified as a SARS-related virus, more specific, the SARS-CoV-2. That's the one that caused the COVID-19 outbreak. And then it's going to show you the genome of the virus. Again, it's around 30,000 base pairs. That one was 29,820. And then it will create a phylogenetic tree. And this phylogenetic tree, just to ground what I talked to you in the beginning, yeah, has a number of clades. The clades on the top, on the red one, is the SARS coronavirus. The purple, the orange, and the green, these are bats viruses. So can you see how you have a number of bat coronavirus, and then one of them jump to the human population? Here in the bottom, the big clade is the SARS outbreak that happened in 2003, 2004. So again, this virus is a new introduction from bats into humans, similar to what happened with the first SARS outbreak. Yeah. So you can also visualize the tree in a number of ways. It's going to become clear from our work and also with Francesco that we take quite serious visualization of data, because a way especially to transmit information to the general community. Again, a new sequence is always clear identified with the right virus, and then with great information. Quite important it is that our tool and others also identify all the mutation on the genomes. So this is here identifying how different is this new SARS coronavirus 2 to the original SARS virus, and that have only 79% identity in the genome. What it means that 23,621 mutations are exactly identical between the first SARS outbreak and that one. So it's quite close, but still have 21% difference. As you understand, is a second introduction into humans from bats reservoir. And then it's gonna go through all the different proteins. And one interesting thing is that some proteins are much more similar between the two SARS strains, yeah, and other ones a little bit more distance. One important information that this software is also going to do is that it's going to identify you all the mutations in every single protein. And that becomes quite important for the scientific community as they try to develop new vaccines and therapies or understand more of the virus. But again, this is the difference between this SARS coronavirus 2 to the first one. How similar are all the sequence of SARS coronavirus 2 in the world? They are extremely similar. They are at the moment 99.9% identical. Yeah. So when people say that there is some mutations about them, it's very small number of mutations, and the scientific community is looking at that every day. So what we know that the virus accumulates a few mutations, but none of them increase transmissibility or pathogenicity. So when you see the news about the virus accumulating mutation, please do not panic, because the virus is extremely stable between all the strains of coronavirus that have been identified in the world, the SARS-CoV-2, they are 99% identical. So this is analysis of one in France against the first one in China. And what you see, it's only 2 mutations, yeah? So from 29,809 positions in the genome to the 29,807 are identical, yeah? And then the software will identify the exactly location of each of the mutations in the different proteins. So one of the big questions that come is, but does this virus come direct from bats and humans? Yeah, and that's why genomics can be very useful. So there was 2 theories put in very early. The first one that came by snakes, selling that market, and then after by Pangolin. So our software or classical phylogenetic analysis can get a look on that, yeah. But what was quite important is that any kind of wrong results have been refuted very strong and very quick by the scientific community. It's a very dangerous time to spread wrong scientific results. This paper on the snake origin, it was retracted with 2 days, because it was quite clear that the results were wrong and the scientific community reacted very, very strong. So that was a paper that was released around like 23 of January, and by 20 suggesting that snakes were the right reservoir. And 1 day later, it was refuted by hundreds of high-level scientists. So here is a tweet from Andrew Rumble. With respect to the novel coronavirus, there is no plausible evidence that the virus has a snake species as it is non-human host. So that was the first one that was in the news, was refuted. The second one that was quite interesting is the pangolin, yeah. So you may have heard about that. And they had some genomes that came from Metagenomics from pangolins in Malaysia, not in China. And when you put that in the phylogenetic tree, it's here, I'm gonna ground you. The pangolin sequence is on the top. The red ones are the human sequence. The referent bat sequence are the purple, orange, and green. And then you could see that maybe the pangolin could be the intermediate roast between the bats and the humans. And that was a theory that was highlighted and was supported for some days. But what you found, if you look for other more close related bat sequence, and here we have the RATH bats from human province in 2019 in China, what you can see is that no, yeah, there was an introduction from bats to pangolin, but was not that one that caused the human, because you have here a much closer bats. So the answer here is that we don't know yet what was the animal that introduced to humans. We know that this virus lives in bats, the natural reservoir. The human virus is completely within the diversity of the bats coronavirus, but we don't know exactly if it went through another host yet, yeah? So our knowledge, it is that it's a bat virus introduced in human. So another software that became completely crucial to this analysis, yeah, it is the software called NextStrain. And that's a software that some colleague of us that developed in Seattle and in Basel, in Switzerland, yeah. And that's a software that's very complementary to our Genome Detective. And the people that developed then, both Richard Newer, Emma, and also Trevor Bedford, they not only they developed the software, but they are really interested on help people to do analysis. And they were here in South Africa in Durban last year, running a workshop with us. So what this software did and why it's so important, this other software, to understand the epidemic in real time. So it's a software that's completely complementary to Genome Detective, as Genome Detective go from the patient to the virus to the sequencing to identification of the right virus, the genome, and the mutations, as I show you. And NextStrain take from there and create big phylogenetic trees, understand transmission around the world, and give reports. So we work quite together with these colleagues from NextStrain, and what they are doing, they are analyzing data around the world in real time. So that's the last report that they have. That's the number of genomes that have been produced for SARS-CoV-2. We had close to 1.6 thousand genomes that have been released in the world. And they have been really important to understand the rate of spread, how it's spread, and now to develop interventions. So I'm gonna take to you quick to some of that. Next slide, we're gonna just see the current update from Europe. So in that phylogenetic tree, what we have now is color-coded the European countries. What you're gonna see from that phylogenetic tree, that's what you call a time-resolved phylogenetic tree. The x-axis is time. So as close to the end, these dots, more recently, they have been sample and sequence. So we are analyzing data, here is March, from like 2 days ago in real time. So what became characteristic from Europe, that was multiple introductions into Europe, with one massive one taking over and being transmitted between European countries, yeah? So what we have is a continuous spread across borders in the last 3 to 5 weeks, yeah, with still a lot of new virus being detected from the very high mobility that they had in Europe with basic without borders and great trains and planes network. That should decrease in the next few weeks as the lockdowns increase. But that's one of the reasons why the European outbreaks spread so strong and so quick, because it was multiple introductions in different countries, very quick, that pick off and start infecting the population. If you go to North America is the same things, many multiple introductions, some of them from Europe. That's the ones on the top here, introduction from Europe to North America. But now what you see is a lot of localized transmission into North America. Yeah, and here was the original like Asian introductions to Europe, to America. Yeah. Transmission across the US is very complex, currently not very well understood, but what we know that one of the first entry points has been Seattle, but now the big outbreak is in New York. We also, what we have in Seattle or in the Washington state is these multiple transmission chains, yeah, with a big one causing the majority of transmissions, but other ones start to pick off. One good thing of the Washington state and Seattle, I work very close with them. I have a position at the Department of Health of University of Washington, Seattle. They have been phenomenal how they respond to the outbreak. Not only Trevor Bedford, it is in Seattle doing this analysis full time, but the laboratory in the university was transformed into a testing laboratory. In 2 weeks, they test. They normally test in 2 days more than the CDC could test in 2 weeks in the whole country. And was a big example on how the academic community could help to respond very quick to that outbreak. If I go to the next one, in Central and South America, we don't have so much genomes at the moment, but what we have is multiple introductions, some of them from Asia, other of them from America, other of them from Europe. Yeah, they are still primary travel related cases in South America. You can see the countries or the province that they come from Hubei, Iran, Lombardy, Italy, China, USA, South Korea, Netherlands, Hong Kong. Yeah, so it's these multiple introductions to South America. I'm especially worried about Brazil. I am originally Brazilian, and we have one of these mad governments, a little bit like Trump and the British one, that first ignore the virus, yeah, and the epidemic is growing very fast. Yeah. So if you go to Asia, Asia was the origin, and that's what you're gonna see here in purple. Yeah, some of that's the closest sequence that was generated in January, as I show. Origin on China. China has done a fantastic job on controlling the outbreak, as some of the other Asian countries like South Korea and Singapore. But what you see that we have been worried at the moment is that Iran had an early epidemic, and now it has recently exported to also around the world, all the way to America and even Australia. And that's why it's important to control all the local outbreaks. Otherwise, you're gonna have this continuous introduction of virus. Yeah. What's in Africa? Yeah, we don't have many sequences from Africa, but that is very likely to change in the next few weeks. Ourselves, the NICD and University of Cape Town is planning, or it's going to be releasing hundreds of genomes to understand that in real time. At the moment, only one sequence from South Africa was produced and released, and that was at the NICD, and that identified a transmission from Europe. But what we have in Africa that's quite interesting is a number of sequences that have been produced in Kinshasa, in Congo. And what they show is already a lot of localized transmission. That's here in the bottom. Different introductions, some from Asia, other from Europe and United States, but already starting to transmit at a high level. In South Africa, this kind of research is going to be really important, especially if we were to start in controlling. We're going to have to do that like a hawk, test, identify transmissions, and try to stop them. Yeah. So what we are doing, we have partnered with CAPRISA. CAPRISA is the organization also inside the University of KwaZulu-Natal in the same building as us, NHLS and ICD. We have recently been funded by the Department of Science and Innovation to answer a very big and very important question. How is the SARS coronavirus spreading in urban and rural KwaZulu-Natal? So we're gonna be doing investigations both in informal settlements, very dense and populated in Durban, but also in some rural area, because it's going to be very important that if we identify, you control that. what we're gonna do, we're gonna use the same way that we have been working with CAPRISA for a number of years. We basically generate sequence data together with epidemiological data to understand cycles of transmission and derive interventions. We did that very well on HIV, understanding who infected young women. And what was good to work together with epidemiology and genomics is that some of our other results, at least on HIV infecting young women, which use exactly the same techniques, influence both international and national policymakers. So we are really working around the clock. Our laboratories are fully open, working with CAPRISA to try to understand that now for coronavirus in real time. Yeah. So to conclude, traditional surveillance and publication systems take too long to respond to outbreaks. Normally, if you take the traditional route, you would not have understand almost anything. Yeah, the speed of discovery of this virus has been a half mark of the scientific community to the crisis. And how they respond so quick is because this was the first virus that's causing outbreaks that we don't know. We had Zika, we had the yellow fever. During that process, we learned how to work together and share data quicker. By the time coronavirus come along, we already knew how to work faster. So that's some of the words that was highlighted that's on the interview that I did with the Nature reporter. Yeah. So I'm gonna take some questions. Yeah, but as I take questions, I would like to leave this slide because that's a big important half mark. Yeah? It is big groups working together. And we are very glad to be doing this big data consortium with Francesco and his team. We also participate in the UKZN Coronavirus War Room with Richard Lasso and big collaborations with other research centers here. And in bold is the people of our group that really are in the laboratories full time, yeah, generating data to understand that in real time. Thank you, Francesco.
Thank you very much, Tulio, for the very interesting and informative talk. Tulio, if you want to answer the questions, we have two options. You can click on the question and answer button and quickly browse what people have asked during your talk, and you might choose to answer some of them, or otherwise I can try briefly to summarize what some of the questions were.
Okay, yeah, let me go answering some of them. Yeah. The first, yeah, the first of one is about Chimera that have seen on TV that swabs for tests are not being taken properly, only from the tongue, not from the back. Are these people trained well? Yeah, that question is much better to our clinicians, Richard Lasso, but I can answer because we are doing a lot of testing. Yes, the sample has to be taken very well. And they have to be taken not from the tongue, they have to be taken really from deep insertion on the nose until it gets to the back or from the back of the throat. It's very, very, very important that samples is well taken, otherwise we risk to have false negatives. Yeah, but what people is healthcare workers are well trained to do that, and it is worrying, but normally people is well trained and people have been doing that and quite well. Yeah. The next one, also from Kimara, why a sub-regional data in each province and clear age distribution not well shown in South Africa? That's not true. The provincial data is very good. We know very well the rate of growth in the different provinces. Yeah. We're going to release a paper today and results showing that the rate of growth was much faster in Gauteng and the Western Cape, the very populated province, yeah, followed by KwaZulu-Natal. But what you see is starting a level of the transmission of data. In relationship to the age and gender, yeah, this data is still quite limited, but I think that the NICD is really working hard to make sure that data is distributed very quick. Again, it's really important. Data, open data, good quality data has been what have helped to respond to this outbreak in real time. Yeah. Yeah, there is a report that suggests the number of tests done in South Africa in the last 4 days have halved. Yeah, we did analyze that data. That's not really true. It is around like 2,900 tests being done in average a day. Yeah, Maria can answer that stronger than me. but we do have a massive lack of reagent kits around the world. This pandemic is happening everywhere, but South Africa has scaled testing very quick and very fast, both through the private and the public hospitals. By last week, they have been done more than 40,000 tests. And the first case that we discovered was on the 5th of March. We have to scale much bigger, but South Africa have initially done quite well in the scaling of the tests. Yeah. How can I get a copy of these slides? I think that you're gonna get better than the slides. You're gonna have this video online. Is that true, Francesco?
Yes, yes, definitely. We'll try to put it online as soon as possible. Yeah. And we can share the slides as well.
Yeah. Next question. There is a version of this trajectory controlled by the number of tests done in the country population. Yes, there are. We're going to highlight that in South Africa, but there is a great resource that's Johns Hopkins Coronavirus Center that highlight that in real time to give idea what may be hidden and less well tested in communities. Yes, that's an important question, but I believe that they are starting to do community-wide testing in South Africa. That's what our health ministry suggests, and it's a very good idea. We have to really understand what is the level of transmission, especially to identify people and stop the trends of transmissions. Yeah. Are there really 3 strains considered the reference as a single base pair difference with possibly no functional difference in the mutation? I think that it's a little bit of misunderstanding there. The 2 different SARS strains, they do have 21% divergence. They are completely different introductions from bats into humans. Between the humans to human transmissions, the maximum that we saw is around now 25 mutations. At the moment, we only call SARS coronavirus 2. We don't call subclades of that. These mutations haven't been associated with any different biological difference, but this small number of mutations is very important to trace the virus, as I show you on the phylogenetic trees. Yeah. Is this a man-made or mutation? It is very clear this is not a man-made virus. Was a very strong publication showing that it's completely in the middle of the diversity of the coronavirus. It's not a man-made virus. It is unfortunately a virus that live in the natural reservoir and that get transmitted. One thing to people to understand is that most of our diseases come from other animals. And there's nothing strange about that. TB comes from cows. Influenza every year either come from pigs or from avians, different species of birds. HIV come from simians. It's completely normal diseases jumping between hosts to humans. It's not a man-made virus. So Kimara asked too many questions, so I'm gonna jump a little bit. I'm gonna go to Richard Lessels. I think. So his questions: why do we not have more sequence data from South Africa if we have 1.4 thousand cases? So I believe that more sequence data in South Africa will come a lot in the next few weeks. What we had, the NICD did a great job in the first one, but they're using a very complex protocol called Metagenomics. This week we send then all the primers that we had here for whole genome sequencing that allow that to do much quicker and much faster. And we're gonna have 3 main sequencing centers in the country that have been funded by the government. That's gonna be the NICD, ourselves in KRISP, and another one in Cape Town with Caroline Williamson. So we're gonna have more data coming very soon. Does the weather have anything to do with the fast or slow spread of the virus? Should South Africa expect the virus to spread fast and wide as we got into the winter season? That was a theory that warm weather would be kind of protected. I think that this theory doesn't hold anymore. We have massive outbreaks in Brazil. Brazil is a tropical country. Have a massive outbreak in Australia. You have a massive outbreak in Indonesia. and it's what is in another one in Thailand. What's going to complicate it in our winter months that we're also going to have a flu season. So it's going to be more difficult to diagnose what it is flu, what it is the SARS coronavirus. But I think that's key is what the government is doing now. It is really try to control the epidemic on the beginning and now increasing tests widely to try to identify the case. Yeah. What could be the possible reasons why we see fewer deaths in the African countries despite an increasing number of infected people? Yeah, unfortunately, the deaths will come. Yeah, in the African countries. It's just that take time. Yeah, the first case that we detected was in the 5th of March. It take around like 20 days or between around 10 days to develop symptoms, and then another people staying in the hospital for another 10, 20 days. So the deaths will grow in the African continent. It's just a function of introduction and time. Yeah. Kimara, she loves asking questions. Yeah. Any information about the development of the vaccine? We have hundreds of groups work on that full time. Yeah. But we expect a vaccine normally take at least 18 months to be developed because it has to pass through development and clinical trials. Yeah, they're going to be doing small clinical trials during the pandemic, but we don't think that we're going to have effective vaccine before we control, or hopefully to control in the next few months. Yeah. Okay, so just going to jump a little bit. Anonymous attendee. In the case of coronavirus 19, which is a new virus, when identifying the mutation, which genome do you use as reference? Do you use the first sequence COVID-19 genome as the reference in this case? Yes, the answer is yes, you are correct to point that. What we did as the scientific community, the first genome, the one was what became a reference genome. And that it go to a database that's called the RefSeq database. Yeah. So when people identify mutations, they always compare with the right genome. So you can compare apples with apples. Yeah. So hi, my J. Why is coronavirus 19 significantly more contagious than original SARS from a genetic point of view? So the scientific community think is that it has an extra protease site that may increase transmissibility, may not. Yeah, I think that we're going to need some time to understand that fully. One thing that changed between 2002 and 2003 to now 2020, it is global flight and communication, how people move. So we are not completely sure if the virus is more transmissible, or maybe that is just an effect that in the last 18 years, we had at least growing air traffic by at least like 1,000% between countries. So introduction of strains are much quicker in new population.
Tulio, maybe one last question, because I'm aware that we can't answer all the questions that have been posed. You want just to choose a random last question, and then we round up.
Okay, so anonymous attendee, the last one. Genetically, what makes SARS coronavirus 2 spread faster than Zika and previous SARS? So what is important, it is that why SARS coronavirus 2 transmit much quicker than Zika is because Zika you need is what you call arbovirus. You need a mosquito. You have intermediate roles there. So the virus, if someone has a virus, mosquito has to bite that person and bite someone else. So not everywhere else have the right mosquito. South Africa is quite lucky, not that many mosquitoes to transmit. Where the SARS coronavirus, as you know, it's by droplets or by people touching surface. So the way to transmit a respiratory virus is much faster. Compared with the previous SARS, I hope I have answered that. We are not completely sure it's more transmissible. we believe that's around the same, or maybe a little bit more, but we're going to need time to understand that. And the biggest difference is a lot of the population movement around the world.
Yeah, Tulio, thank you very much. I think I can thank you on behalf of 205 people that listened to your talk and viewed your very nice slides. So a very big thank you from all of us. Some people asked, will we share slides and video? Of course we will. We will try to put the video like we did last week as soon as possible on YouTube, and we will make a plan to share the slides as well, and we will let you know soon. Yeah. Also, we will try either today or latest on Monday to tell you who our next speaker will be next Friday, so that we will continue this new tradition of Friday mornings breakfast talks. So again, a big thank you to Tulio, and a big thank you to all the people behind the scenes that made this webinar possible. Ilya Kimara and the UKZN ICS team, and I wish you a very good and safe and healthy day. Yeah, thank you very much. See you next week. Bye bye. Bye.